So this chapter is on the chemical building blocks of life, which is carbon compounds. And so we're going to focus here on the biomolecules that make up cells that you find in your body or other living organisms. And so the first section is going to focus specifically on carbon as the basis for these molecules. And your learning outcomes here are going to be to describe the relationship between functional groups and macromolecules. Certain molecules are going to have these groups of atoms on there called functional groups, so we'll take a look at that. And these functional groups are characteristic of those types of molecules. And then recognize the different kinds of isomers. And so we'll define those and see what those look like. And then list the different kinds of biological molecules here. And I'll point out these learning outcomes here as before. They could actually serve as ways to self-assess. When you look at them, they look like they're essay-type questions. So you can go back and then see, can I answer those and recreate some description of what these learning outcomes are asking me to do. But this is our goal. Some of the basic goals there. So when we look at the biomolecules, the molecules of life, they're basically connected to the chemistry of carbon. Carbon itself has 4 covalent bonds, and carbon can bond with itself and form long chains. It can bond with other kinds of nonmetals like oxygen, nitrogen, sulfur, phosphorus, and hydrogen. And we want to recall from our prior chapter here that all of those elements that we find in the first column have 1 valence electron, and the valence electrons are the ones that are involved in chemical reactions. Second column is 2. We come over here to the other part of the main block, sometimes called group A. These have 3, and then there's carbon right there with 4 valence, and then we have 5, 6, 7, and then 8. An exception is helium, which only has 2. But the point is carbon has 4 valence electrons. And so if we draw the dot structure there, we see that carbon still needs to have 4 more electrons to build that octet. And so what it can do, one of the things it can do is it can bond with other carbons and form covalent bonds. And everywhere there's these single electrons, we can form more bonds, so that you might bond a hydrogen here and form a covalent bond there. And if all we have is a bunch of carbons, each dash is a pair of shared electrons, then we need to bond them to other things to give each carbon an octet. So each dash is a pair of electrons, so that's 8 around there and so on. And the simplest organic compounds we can make are these hydrocarbons. They're called hydrocarbons because they have carbon and hydrogen. Okay? And so we might remove these and add other things like oxygen and nitrogen and so on. And then you're going to start to get compounds of different properties. You could also instead start branching off. So you can have these chains of carbons. You can form rings of carbon. And so the diversity of biomolecules are really truly based on the fact that carbon can bind with itself and form those 4 covalent bonds. So I mentioned hydrocarbons earlier, and I had drawn one here with 1, 2, 3, 4 carbons right here. And those are pretty much nonpolar molecules. The thing about having a polar molecule like water is, number 1, you have to have polar bonds, which was discussed in the prior chapter, and number 2, the molecule has to be asymmetrical, so that there's an off balance of negative charge throughout the entire molecule. Now, the molecule is not an ion, it just doesn't have the charge balance, the negative charge balance throughout the entire molecule. But a hydrocarbon can't even be a polar molecule itself, because the bonds between carbon and hydrogen, neither one is very much stronger than the other at pulling electrons towards it. So the bond is considered pretty much nonpolar. So already you can't have a polar molecule. Now you might get that though when you start adding other things and you start changing the symmetry of the molecule. Those other things we might add are called functional groups. And those truly change the properties of the molecule. here giving them different properties, and then allows us to categorize them into things like amino acids and other things. So here are the functional groups, and I'm going to want you to be familiar with them, so that means to make some effort to learn them and where they're found. One is the hydroxyl group, and here you can see a chain of 2 carbons, and there's your hydroxyl group. So it's just OH. It's not the same as a hydroxide ion when we covered water chemistry and acids and bases before. This is a charged particle. It's not covalently bonded to anything. It's not the same. This is the hydroxide ion, which is a strong base, but this is hydroxyl. It's neutral, and that dash means it's bonded to a carbon or some other group of carbon containing a part of the molecule. Then we have the carbonyl. By the way, the hydroxyl is found in carbohydrates, proteins, and nucleic acids. And then we have carbonyl. And there you have a carbon with a double bonded oxygen, and then you might have more carbons out like this, or hydrogen out there. And here you have acetaldehyde, and there's your carbonyl group there. And we find these in carbohydrates and nucleic acids. And then we have the carboxyl group. And this carboxyl group, these are the structure, how they look structurally, how the atoms are somewhat arranged relative to each other and bound to each other, bonded to each other. Sometimes be familiar with the idea that you may see a further shorthand of carboxyl as COOH. And the first O is this one, and then the second one is the part with this group right here. Now, this is not hydroxyl. We're talking about this being the entire group to be your carboxyl. And in this case, they've given us an example of a very simple organic acid called acetic acid. So we find these proteins, lipids, and then these organic acids. You might have heard of citric acid. In lemons, they're going to have this carboxyl group. Amino acids have this carboxyl group. And the reason this is the case is because for the carboxyl, this H here, when you put whatever the rest of the molecule is, I'm just going to put R for the rest of the molecule. This one hydrogen can pop off, leaving its electron behind and become a proton, and that makes it a weak acid. Now, if I have citric acid and I have a billion of those citric acid molecules in solution, there's going to be a very, very small proportion that ionizes like this and produces the protons. It fits the definition of a weak acid, like when we were studying buffers earlier in the prior chapter. And then we have the amino group, and the amino group is a nitrogen with 2 hydrogens. And sometimes you're going to see it shorthand, so make note of this, be familiar with this. You might see it as NH2. Okay, so if I have a molecule, some carbon molecule R, and then I see NH2, you recognize that as being amino, same as if you saw it drawn more as the way it would structurally. Like this. So these two are the same thing. That's amino, that's amino. And of course, we're going to find these in proteins where you find amino acids. And they give an example here with an amino acid called alanine. So found in proteins, we also find amino groups in nucleic acids, which are DNA. The Na stands for nucleic acid. And then sulfhydryl, this is sometimes just written as SH. You can see that bonded there, and you find these in proteins. There are some amino acids that have sulfur in there. So proteins will have sulfur in there because of amino acids like cysteine. And then you have your phosphate group right here, and your phosphate group is right here, bound to the rest of the carbon molecule here. It's going to be found in, for example, nucleic acids. And notice that it's a phosphate with 4 oxygens, and this one oxygen that is bonded, just like it is right here, to the rest of the carbon there. Sometimes you'll see it shorthand as PO4 with a 2- charge, and that would be your phosphate. And then we have maybe looking at a very simple molecule called methane. This is a structural formula. This is the molecular formula, CH4. If I remove this right here and then attach this to another carbon, then we can call this a methyl group. Like methane, but methyl. So sometimes you'll see it as written as CH3 with a dash indicating a covalent bond to the rest there. And here you see there's your methyl group right here attached to another part of the molecule. In this case, it is an amino acid called alanine. So amino acids make up proteins. So make sure you learn those to commit them to memory. You're going to see them in some of the chemistry we're going to be covering as we go through metabolism and the mitochondria and we go over DNA replication and things like that. So next thing we want to look at is isomers, and isomers are going to be molecules with the same molecular formula, but the structures are going to be different. Okay, so an example would be a 6-carbon sugar like glucose, C6H12O6. It turns out that glucose, which is one simple sugar with this formula, it turns out there's other simple sugars that also have this exact same formula, but the molecules or the atoms are arranged in different ways. Fructose, galactose. They have the same formula, C6H12O6. So they would be considered structural isomers. And then there are stereoisomers, which are structural isomers. And one of the ways they differ is that if you take one part of the molecule and you just arrange it slightly different on the carbon atom, you've created a mirror image of the molecule. It seems a little bit weird, but you want to think about your hands are mirror images themselves, so they have the two here. This would be, for example, your right hand, and this would be your left hand. If someone was facing you and they held their hands up in front of you like that, then this is the right and this is your left hand there. And you can see here on the carbon that the Y is here and then the Y is here. That's mirror image. If both hands were exactly the same with the thumb out here, then they would not be these stereoisomers, sometimes called enantiomers. This Y would actually be over here and the Z would be over here. And the X would be over here and the W would be over here. In this case, they would not be these stereoisomers. Now, they are given a D and an L for the way they behave when certain light shines, and they call, well, this is right-handed and this is the left-handed version. The D stands for dextro, which means right, and then L levo for left. And the phenomenon of chirality is just that the chiral molecules is the way light interacts with them. The light gets bent a different way depending on which one you have in solution, when these particles are dissolved in solution. The point here is a big one here. There's a difference between a D sugar and an L sugar, and an L amino acid and an R amino acid. They would be mirror images similar to this. And the big significance here is that you have enzymes that interact with and bind with these molecules. Enzymes are what runs your metabolism, and those enzymes are very specific for one form or the other. Okay, so you have enzymes that interact with D sugars. They're not going to be able to bond with L sugars. So these enzymes are very, very specific for the kind of molecules that we're talking about. This is a diagram here of, or a table of the types of molecules we're going to run into in this chapter, and so you want to make sure you go through it. And the big idea here is when we look at macro, that means large. Micro means small. And macromolecules are going to be basically, in many times, they're going to be made of smaller subunits that are similar, not exactly the same, but similar, called monomers. Okay. And when you start putting these monomers together, they're going to make larger molecules called polymers. Okay. And so poly means many. And so this is just a table summarizing some of the major categories of biomolecules. There's 4. There's carbohydrates, nucleic acids, proteins, and lipids, right? And so the macromolecules include starch and glycogen. Starch is how plants store sugar, glucose, and then glycogen animals. And then there's cellulose. And the thing about these is they're made of glucose. And the difference is in the way the glucose molecules are bonded together. They're bonded together differently. Starch and glycogen are used primarily for energy storage, but cellulose is used for structural support. It's what makes plant cell walls. Okay. And then there's chitin. Chitin is a special modified glucose molecule, and that we see in the exoskeletons of crabs and insects. And then you have nucleic acids. The DNA stands for nucleic acid. And RNA, the NA stands for nucleic acid. One is deoxyribonucleic acid, the other one is ribonucleic acid. Their monomers are called nucleotides, which we'll see in a bit. And this is basically how genetic information is stored and then used to make proteins, right? So the DNA codes for proteins, which is the major molecules that run everything and build structure and so on in your body. So DNA encodes the instructions, And then the RNA is the go-between. The RNA is what takes those instructions to the machine called a ribosome to make them. So example would be messenger RNA there. DNA is found in your chromosomes. And then proteins, those are made of monomers called amino acids, and there's functional and structural ones. Functional ones typically are going to be dissolved in fluids, and they are enzymes that facilitate reactions, speed up reactions, or in transport, so they'll carry things like hemoglobin carries oxygen in your blood. Structurally, they might form things like hair or spider webs made of silk. And then we have lipids, and then lipids include things like fats and oils, or they're also called triglycerides, phospholipids, which make up your plasma membrane, and then some others. Probably you've heard of steroids before, for sure. And so the lipids are a little bit more diverse structurally. There's a big difference between the way fats look and steroids, but one thing that all of these do have in common, the lipids, is that they are nonpolar molecules. And so just a little table here showing some examples here. Here you have a chloroplast here. A chloroplast uses sunlight to help generate carbohydrates, simple sugars like glucose. And then the glucose bonds through with the help of enzymes to make a 2-carbon sugar. And then those 2-carbon sugars can be transported, say, to the roots where the glucose molecules are bound together to make a large molecule, a polymer called starch. And every one of those little ring-like structures is one of these glucose molecules here. And then nucleic acids, they get their name because they're found in or originate in the nucleus. They would make up structures like chromosomes, and DNA is part of that chromosome. So here's your polymer here, and then the monomer is your nucleotide. And so you have these repeating units of these nucleotides. Just like over here we had our monosaccharide making this larger polymer. And then we have proteins, and this one looks like a structural protein, looks like part of the cytoskeleton, the skeleton inside of a cell. And you see these long filaments here, structural filaments. And if we zoom in, they're going to be made of different kinds of amino acids. And that polymer is called a polypeptide. We'll see why later. And each one of those, or these subunits, are just basically variations of amino acids. And the way they differ is going to be in a functional group that's attached there, a group of elements. So this one happens to have a methyl group, but other amino acids will have other things there. So they'll vary right here. Sometimes we'll call that R, the variable R group. And then we have lipids. And the one lipid they have here, the ones that we store in our fat cells, they're called triglycerides, which are fats. And if they're liquid, they're oils. And you can see here they have one type part of the molecule here, but then they have these repeating chains of hydrocarbons here. And hydrocarbons have a lot of energy stored in them. There's a lot of energy stored in the carbon and hydrogen bonds. And so this is your basic monomer, which is a fatty acid that helps make your triglycerides. Now, in order to make larger molecules called polymers from subunits, it's always going to be done by this process. So you want to become familiar with it. It's called a dehydration synthesis, which means your molecules are going to lose the equivalent of a water. Now, you're not taking a water that's on the molecule and removing it. You're taking out parts of two molecules that, when they're removed, those two parts combine to make water. And so we have a very simple example of this here, and we'll see specific ones later. And here, these spheres just represent part of a monomer. Okay, so monomer might be like glucose, for example, which is really C6H12O6. And on part of that monomer there, you're going to have functional group like hydroxyl. Okay, we saw that earlier. So here we already have two monomers joined together. This is a dimer, and it's going to react with a monomer over here. This is the third one here. And the way they're going to do that is we're going to remove an OH from one molecule and an H from another. Those two go off and form water, which is called dehydration because you're losing what's equivalent to a water. And once you lose that here, then a single covalent bond is formed there. and we've made a larger molecule. So this is called a dehydration reaction because water is lost. So remember, dehydration makes bigger molecules. So we get larger and larger molecules called polymers. Usually these dehydration synthesis sometimes are called condensation, like when water condenses from water vapor. Condensation reactions. And a thing to remember about this is that this process of building, like making a large building, requires energy. Okay? So requires that energy there. Now, if we just reverse this and went backwards, basically that's what we have here. Those are going to be called hydrolysis reactions covered here. Lysis itself means to split. We're going to see this term again. I'm going to keep reminding you. What does lyse mean? To lyse means to split. And when we translate hydrolysis, hydro means water. Literally translated means water, using water to split, or water splitting. And so it's basically the opposite here. If you lost water to make a larger molecule, what you're going to do is you're going to bring water in, and you're going to use water to split. So basically we're going to break water into OH, a hydroxyl group, and an H, and then use it to break that bond right there. And so one of the hydrogens here goes and bonds to this part of the molecule over here, and there it is right there. And then the remaining OH, or hydroxyl, goes and covalently bonds here, and we've split the molecule into two smaller parts. This is called hydrolysis. So keep this in mind, something to know. By the way, as we break this down, as you're breaking things apart, that's going to release energy. Okay? So this is part of our metabolism. We build and we break down. We build and we break down. When you're building, usually that's called anabolic or anabolism. And anabolic building requires energy, and breaking down would be catabolic or catabolism. So this would be catabolic reaction or metabolism. So the two combined are all part of your metabolism. So this section is on carbohydrates, and for carbs, they are for energy storage, and they also help provide structure. And so your learning objectives for our carbohydrate section is to describe the structure of simple sugars with 3 to 6 carbons. So there's you have sugars that have 3 carbons, some that have 6 carbons, some have 5 carbons. And then you're going to relate the structure of polysaccharides to their functions. So when we look at carbohydrates, the simple ones, are going to have a ratio of a 1 to 2 to 1 of carbon, hydrogen to oxygen. So this would be what we were talking about in the last section, an empirical formula, which is the lowest ratio that you can get. So that's understood to be 1, so that would be C1H2O1. And then the N on the outside of this empirical formula, you would put a number there based on the number of carbons, and then you just multiply the subscripts by that. So if this is a 6-carbon sugar, then this would be 6, and then you would multiply everything in there by 6, and so then that would give you C6H12O6. If this were a 3-carbon sugar, then this would be a 3 here instead, and then the answer would be a carbon with C3H6O3. If it was a 5-carbon sugar, then it would be C5H10O5. Okay. Now, a lot of carbohydrates as an energy molecule, the energy is tied up in the electrons associated with the carbon-hydrogen bond. So anytime you have a carbon organic molecule with a lot of hydrogen, that's where the energy is. It's going to be in the hydrogen that has the electrons that it brings to the relationship. So that's the idea there. The more hydrogen, the more energy. So when we compare later carbohydrates to fats are going to have even more hydrogen in there, so they have even more energy. So carbohydrates are going to be a good energy source, and examples include sugars, starch. Glucose is a very specific kind of sugar. Sugar is more general. Starch is a polysaccharide. Monosaccharide. Saccharide is, I believe it's ancient Greek origin, which means sweet or sugar, and then mono just means one. So these are going to be the monomers. 6 carbon sugars do play a lot of important roles within the body. One very common one is glucose. Already mentioned earlier, C6H12O6. Fructose is going to have the same formula. It's a structural isomer. We'll take a look at that. And then galactose is an isomer as well. It's a stereoisomer, just where you're going to have sort of a mirror image, at least on part of the molecule there. Now, there's enzymes that work on very specific sugars. So the enzymes themselves, they work to break down or build up larger molecules of sugar or break them down. They recognize very specific forms of C6H12O6. So they're specific for those different structural or different types of sugars based on their structure. So if there's a sugar, there's going to be an enzyme for it. So when we look at your basic sugar, we can number the carbons on there. And usually it's done from wherever there's a major functional group. So when we look over here at the end, you have this double bonded oxygen here. And so that's significant. So we're going to call that carbon number 1, and then carbon number 2, 3, 4, 5, and 6. Now, if you put these sugars in water, so this is what the molecule looked like without being in water, but when you put them in water, this part of the molecule here is going to react with this part of the molecule here, and a little rearrangement of the atoms is going to take place. So you can see that is taking place right here. As we put it in water, you can see that this interaction is going to occur right here. And you're going to end up with 2 possible forms. You're going to end up with and it forms a ring-like structure. So you're going to end up with what we call alpha glucose and beta glucose, and it depends on where this hydroxyl group ends up. Like here you see it toward the bottom and here toward the top. And you can still count the carbons. This is the number 1 carbon, 2, 3, 4, 5, and then number 6 is up there at the top. We still have the same arrangement here. It's just that the hydroxyl is in a different place. This is going to become pretty significant when we start looking at when these sugars bond to each other, whether or not we can digest the more complex carbohydrates that you get from there. When it comes to the fructose, the glucose, and the galactose, if you went and you calculated, added up all the carbons, hydrogen, and oxygens, every one of these is going to give you C6H12O6, but they behave differently because they're structurally different. And we even have tools to sense some of these differences. Fructose, for example, is many times sweeter than glucose. And you probably, if you've ever eaten honey, you like it. It's so sweet it makes you make a face, right? Because it's very sweet. But you can see that fructose and glucose are structural isomers. You can see there's a different location here for this double bonded oxygen here, as opposed to being up here at the end. So structurally overall they're different. And then over here we have this sort of between glucose and galactose, this sort of a mirror imaging right here. So this makes it sort of a stereoisomer right here. So if you were to put a mirror here, you can see that the hydroxyls are on different sides, making this sort of mirror image of each other type of isomer. And structurally, they're different. And that would be recognized by the appropriate enzymes. Then we have disaccharides, and these are 2 sugar molecules. So in order to link the 2 simple monomer sugars, you would have to have a dehydration synthesis, which was mentioned earlier. And you can see that occurring here. So here you have alpha glucose, mentioned earlier, and fructose. And the 2 molecules are side by side. We can go and count the carbons if we want. And I want to point out that sometimes when they're drawing structural formulas in organic chemistry, they do a little shorthand. And everywhere there's a bend between these dashes or those lines represent a covalent bond. There's an atom right there, and it's not drawn in. It's understood. When it's not drawn in organic chemistry, understood to be a carbon. So there's a carbon right here. Okay, there's going to be another one right there. So that's 2, 3, 4, 5 right there at that bend, and then one actually drawn right there, 6. So there's your 6-carbon sugar. Here's fructose. We got 1 carbon right there, 2, 3, 4, 5 right there, and then 6. So you can see here that each one of those is a 6-carbon sugar. Sometimes we call those hexoses in general. Okay. And you can see here that the hydroxyl on one and a hydrogen from another both have hydroxyl groups. So the 2 hydroxyl groups, the functional group, that's where the action is going to occur. That's why they call them functional groups. But we're going to lose this OH and this H, and that's going to produce this water. And we end up with our covalent bond here, and we end up with a disaccharide called sucrose. This is commonly called table sugar. This is what we put in our tea, what we cook with. And it's actually a major transport sugar in plant material. So let's say we're going to get sucrose from sugarcane crop. Basically, they're harvesting the sucrose that's in there. Here is maltose. Maltose is a disaccharide made of 2 glucose molecules, and you can see there's that bond. Sometimes they call the covalent bond between the 2 sugar molecules, they call it a glycosidic bond. It's just a specific name for a bond between sugars. There's another important one that is not shown here, and it's between glucose and galactose. And when those 2 combine, it gives you a sugar found in milk called lactose. And it's a disaccharide. So you might want to add that to your list there. We have sucrose, maltose, and another important disaccharide is lactose. And it's between glucose and galactose. And some people have trouble digesting that one because in order for us to digest it, you have to break the glycosidic bond with a hydrolysis reaction in order to get the 2 simple sugars. And what happens there is that individuals stop making the enzyme lactase, which is used to break down lactose. And so you can't absorb that sugar, and then it makes it from your small intestine to your large intestine, and then the bacteria take over, and then the individuals get sick. They may get diarrhea, get a lot of gas, and so on, and it's just not very comfortable. Then we have polysaccharides, and polysaccharides, poly means many, so many monosaccharides joined together, and like any other building reaction, anabolic reaction, is going to be a dehydration synthesis requiring energy. Now, some of this polysaccharides are used in energy storage. When you eat French fries, the French fries have a lot of starch because it comes from a potato, a plant. Animals are going to take those monosaccharides and bond them to form glycogen. So we have glycogen stored in our liver, in our muscles, and all that is just a bunch of glucose molecules just like starch. They're just bonded together for storage. And then you have structural support. Plants have cell walls that help support their cells. The wood has a lot of cellulose in it, along with other compounds. And then arthropods, which are animals that don't have an internal skeleton. They have an exoskeleton that's jointed, like this lobster here, or like a spider or cockroach. Those are arthropods. Their exoskeletons made of a modified glucose molecule bonded into a polysaccharide, and that is called chitin. Chitin is also found in the cell walls of fungi. So while plants have cell walls made of cellulose, which we see right here, fungi have cell walls made of chitin. So cell walls of fungi is chitin. Okay. So here we see how we might go about making starch, okay, and the difference between starch and glycogen. You can see over here that the starches, and there's amylose and there's amylopectin, which has some branching, but basically starch is usually just one long chain. It might coil up like you see here. What it is just a bunch of alpha glucose molecules bonded one after another, and then it coils up into this helical-like structure there. And it says that the linkage is a 1,4. So just a little quick interpretation, just so you say, wait, what does that mean? Well, remember we name or number the carbons, right? And we started, this is number 1 carbon, 2, 3, 4, 5, and then this is the number 6 carbon. Okay, and again, 1, 2, 3, 4, 5, and number 6 carbon. So which 2 carbons are bonded together? This is 1 and this is 4. So it's an alpha 1,4 linkage. So that's what that means there. So these are alpha 1,4 linkages. When you have a 1,4 linkage and that's all you have, then you get just a long chain. And so that's basically the starch. Now in animals, they tend to store the same kind of molecule. It's a glucose molecule, but there's going to be a lot of branching. Now you're going to have the alpha 1,4 linkage to make a long chain. So we're going to continue with more of the molecules coming out this way. But then you're also going to have branching off. And that branching off is going to be a 1,6 linkage. So let's take a look here. This is a 1,4. This was the 1 and this is the 4 carbon. But here we have 1, 2, 3. 1, 2, 3, then this is number 4, then 5, and this is the number 6 carbon here. And then over here, this is the number 1 carbon. So this is going to be an alpha 1,6 carbon, and that gives you the branching off. So the difference between glycogen and starch is that glycogen is going to have a lot of branching off due to the 1,6 linkages as well. And then all we do is just have enzyme to break it down, and then you can use the glucose for energy in your mitochondria to make ATP. In the cells and part of the mitochondria. Now cellulose. Cellulose is the paper you write notes on. Cellulose we find in wood and so on. It's made by plants. And cellulose uses beta glucose instead. Remember, beta glucose is the difference where that hydroxyl is. In alpha, the hydroxyl is down here on the other side here. And so what we have instead is a beta 1,4 instead of an alpha 1,4. And there is a difference here. And one of the differences is that we don't make enzymes that can break that. Okay? The enzymes that we have to break down starch, we don't have to break down, the enzymes break down or break the alpha 1,4 linkages, but no animal makes an enzyme that can break down cellulose. So cellulose is used to make these these fibers that give structural support to cell walls and so on. Now, there are microorganisms, though, that produce cellulase, which is the enzyme to break down cellulose. You can recognize the enzymes because oftentimes they end in ASE. Enzymes are used to get reactions to occur here. So that's a big difference there. The beta 1,4 linkage is way different. We don't have enzymes to break it down. So what does this end up being? This is the fiber in our diet. When we eat, it goes right through us, and it's very good for digestion, and it's very good for making sure that you have stuff moving through you smoothly. In section 3, we're going to be covering nucleic acids, and the nucleic acids, the name is derived from the fact that these molecules originate or are located in the nucleus, and they're associated with storing information. Genetic information. So thinking about DNA, the Na stands for nucleic acid. So we're going to be looking at the basic structure of the monomers called nucleotides, and then look at similarities and differences between DNA and RNA, and then their functions of DNA and RNA, and then recognize some other kinds of nucleotides that are involved in transferring energy, which would be an entire other chapter when we get to all those times we talked about mitochondria being the powerhouse of the cell. In this case, we're going to look a little more closely at these molecules that do carry energy. Now, we call these nucleic acids. That's the name of the polymer. So when we talk about DNA, it is a polymer of a general category called nucleic acids. The monomers of this polymer are called nucleotides. So let's go ahead and sketch your basic nucleotide, which is going to be made of a sugar, a phosphate, and a nitrogen or a nitrogenous containing base, or a nitrogen containing base or a nitrogenous base, they call it. Now the sugar itself is going to be a pentose, and penta means 5. In the last section, hexose has had 6 carbons. So this pentose is going to be one of 2. It's going to be either ribose or deoxyribose. And if it's DNA, it's going to be deoxyribose. And deoxy means without oxygen. It has 1 less oxygen in its structure. But let's do a little sketch of what a nucleotide would look like. We're going to draw, and I recommend sketching some molecules at least once. It's a form of kinesthetic learning, that sketching. What we're going to do is we're going to draw an oxygen right there. And then remember, right, everywhere there's a bend, it's understood to be a carbon. So there's a carbon right there at that bend. There's going to be a carbon right there at that corner. And then another carbon. So right now there's 1, 2, 3, 4 carbons. Okay, this is going to be your 5 carbon sugar, except we've only drawn 4. We still need 1 more. So from this carbon, we're going to draw another covalent bond, and we're going to put a bend right there, and that bend right there is the 5th carbon. Okay. Now to this carbon right here, we're just going to draw a little square for now, and then put an N in there, and that's your nitrogenous base. And then one of the functional groups that I asked that we become familiar with in the prior section is the phosphate group. And so here is where your phosphate group is. That's a P right there. Okay. So this is your nucleotide. Okay. And the difference between, there's a couple of differences, there's a few differences between DNA and RNA. But one of the differences, it has to do with the name, deoxyribose or ribose. Right here we start right around where the first carbon is, where the nitrogenous base is bonded to it, and we go downward to this next carbon right here. At that carbon, if we're missing an oxygen that's usually there, then that would be a deoxyribose. If you have the oxygen there, then that would be the ribose. I'll show you in a picture a little bit later. But the nitrogenous base actually is a larger ringed molecule that has carbon and nitrogen in it, and it has some weak base properties to it. And so those positions of those atoms in there are numbered. So when it comes to the sugar, instead of just number them 1, 2, 3, we're going to say 1 prime. So this is the 1 prime. And so we're going to write 1 prime like this, 1 with like an apostrophe or a little mark like that. This would be the 2 prime. So it's at the 2 prime carbon that we have the missing oxygen for deoxyribose. Ribose would have the oxygen there. This would be 3 prime. We have 4 prime. And then 5 prime. So sketch it and then see if you could resketch this kind of thing again, from memory, right, when you self-assess. Now, the way this works is that this molecule has an orientation to it. It has a top and it has a bottom to it. And my point here is that the side of the molecule where the phosphate is located is the 5 prime side. And on the opposite side is the 3 prime side. That's very important because when we have the enzymes that build these molecules, they're always going to add to the 3 prime end. So kinesthetically, I recommend that we sketch at least 1 as we're drawing. So I was trying to ask you to say, go ahead and draw along with me. When you draw like this, we're going to put another phosphate there. Remember, the dashes represent a covalent bond. So this is a covalent bond between the phosphate and the 3 prime carbon of the sugar. And then we're going to repeat the bend for the 5 prime carbon. There's the oxygen. And then another nitrogenous base here. And so what happens here is, if we're going in this direction here, we're going from the 5 prime to the 3 prime side of this growing molecule, and the enzymes are always going to add to the 3 prime. That's something to remember for the future when we cover how DNA replicates itself. Now, if it's DNA, there's going to be 2 strands, and the other strand would be going in the opposite direction for DNA. Okay, so you would just flip these molecules upside down, and the nitrogenous bases would be facing inward, and the bases are actually going to pair up with each other. There's pairing rules that they follow, and we'll go over those in a little bit. Okay, so now DNA, that's one big difference. DNA is double-stranded, so DNA would be going in the opposite direction. And so the other one would be going in the 5 prime, 3 prime. It's very important to remember that when we go over DNA replication later. Now, the two strands run parallel to each other, but in opposite directions. That would be referred to as anti-parallel. The anti-for they're going in opposite directions. So, again, it depends if DNA's, the difference is deoxyribose and then ribose for RNA. Ribonucleic acid, deoxyribonucleic acid. Now, when it comes to the bases, in DNA, there's 4 kinds, possible kinds. And then RNA has a different kind of base when we compare it to RNA, when we compare it to DNA. Uracil is found only in RNA, and thiamine is found only in DNA. So this one is DNA only, and this one is RNA only, and the others are found in both. Okay, so let's write only and only. And sometimes we just refer to them as A and T and C and G and U. They're in categories. They put A and G, adenine and guanine, in purines. And here, I remember these mnemonic devices. I had a student who was studying out loud, and he's studying another student, and he says, "Hey, all you got to do," and I'm overhearing him as he's peer tutoring. He said, "All you have to do is remember girls are pure." And I stopped and I'm like, "What are y'all talking about?" And he was like, "Yeah, that's a way to remember it because the girls has a G in it, like guanine, and then the A for adenine, and then there you go, girls are purines." So that was his trick of remembering it. And then the others are the pyrimidines. Okay, and so that would be thiamine and cytosine and uracil. Now, the bonds that occur, if we go back and look at what the phosphate groups looks like, remember the phosphate has 4 oxygens around there, so this is a group of atoms that has phosphate in the middle, so that means there's an oxygen on both sides of the phosphorus on either side, and anytime you have that oxygen then connected to a carbon on those sugar molecules, they're called, it's a kind of bond in organic chemistry we call ester bond. And so it's a phosphodiester bond because you have 2 ester bonds on either side. So that's what this term means, phosphodiester bond. It's just a special kind of covalent bond, just like in sugars you had glycosidic bonds. And when we cover proteins in the next section, the covalent bond between amino acids is called a peptide bond. Right? They're just special kinds of bonds. So looking now at the diagram from your book of a nucleotide, we see what our drawing is better, even though this one shows the detail. You don't have to memorize the complete drawing here, just be able to have an understanding of the basic setup here. Here's your nitrogenous base coming off to the side there of the 1'. This is the 2' where you're either going to have an OH here if you're DNA, or just an H there if you're RNA. And an OH of your RNA and an H of your DNA, because the DNA would be missing the oxygen there. Okay, and then there's your 3', 5', 4', 5' carbon, and then here's your phosphate group. And those are the oxygens I was talking about that are involved in bonding. Actually, it's this one right here. This is an ester bond. That's the ester bond right there, and then we'll have another one formed to the next molecule there. So here's what it looks like here. Here is your 5' right here and your 3'. This is just one strand, and they're not distinguishing between DNA or RNA. We have the nitrogenous bases sticking out to one side there, and sometimes they call this part right here, because it's alternating between phosphate and sugar, they'll call it a sugar phosphate backbone. It's like your backbone supports your body, right? And then the nitrogenous base out to the side. Now here's what the 2 different categories of nitrogenous bases look like. You have your guanine and cytosine. Remember, the girls are pure. They're the purines. The purines actually are double ringed. You don't have to memorize their structures, okay? But just recognize that they are actually, the purines are larger with that double ring structure than the pyrimidines. It's kind of funny that we have a longer name for a smaller molecule. That's kind of the way I remember. Which one's the smaller one? The one with the bigger name, the pyrimidines, right? Adenine and guanine are found in both DNA and RNA. And so these right here, the double rings, those would be purines. Okay, so these would be the purines. Remember, the smaller name is the bigger, and then this one would be a pyrimidine, and it could be any one of these. And so you have the cytosine, thymine, and uracil there. Now, RNA and DNA are like this. RNA, the way we cover it, we're going to cover it. It's only one strand. It may fold on itself. You remember, for DNA, you're going to have another strand running in the opposite direction. Okay, and when you do, there's actual pairing rules. Okay, so these DNA pairing rules, you're always going to find A pairing up with T. And the way I remember that is, if you can draw the capital letters with straight lines, they go together. Okay, and they don't form strong covalent bonds with each other. So we're talking about the other strand, the nitrogenous base that would be out to the side, and it's always going to be a purine with a pyrimidine. If you notice, A is a purine and T is a pyrimidine, so it's always one long one with one short one, or one bigger double ring with one smaller double ring. And then it's always going to be C with G, and we're going to draw not 2 but 3 dashed lines. As you remember earlier, when we were talking about hydrogen bonding, we would draw dashed lines, and that's exactly what that is. There is, like with water molecules that form hydrogen bonds with each other, these nitrogenous bases can form hydrogen bonds or weak interactions. And so while the individual attraction between the A and the T and the C and the G along the long strand of DNA are weak, combined, it provides for a relatively stable attachment between the two strands. That's only for DNA. Okay, so here is some of that right there, and you can see that we have, between the A and the T, they have the hydrogen bonds there between the two strands, and they're running in opposite directions. If you look closely at the sugars, they're facing opposite directions. Okay, one of them is going from in the 5' to 3' direction, the other one's going in the opposite direction. So the base pairing rules are down here, A with T, except with the RNA, because RNA is not going to have thymine, it's going to have uracil, right? So then uracil would be what matches up with A when we talk about some of the processes dealing with RNA, but C always with G regardless. They're going to refer to this a twisted structure as a helix, and when it's 2 molecules, it's a double helix. Okay. Now, what's the big deal here is that with those 4 letters found in DNA, the A, the T, the C, and the G, if it's the sequence of those letters as you run down these molecules that are extremely huge. We're talking about billions of these long. Okay. If we look at all of our genome, all of the genes that you have in one cell, one nucleus, an individual strand of DNA would be hundreds of millions of these long, and it's the sequence of those A's, T's, and C's and G's that it's that sequence that gives the coding for the proteins, and it's every 3 letters. Every 3 letters is a possible A, T, C, or G, and if every position on here is a possible A, T, C, or G, and every 3 gives you a code for building a protein, then how many possible codes are there? Well, in the first position, let's say that it's these first 3 letters here. What are the possible combinations there? You can have an A, a T, a C, or a G. That's 4 combinations possible there. The next position is the same, 4 possibilities, and then 4 more. And so each one, independent of each other, that's a multiplication rule. And so it's 4 times 4, 16 times 4, gives you 64 possible combinations to give codes for 20 amino acids. So the coding is enough to cover the 20 amino acids that we do have that build our proteins. So this code it's really cool. And remember, it's every 3. So for RNA, remember RNA, there's deoxyribose. So we want to compare them. How are they the same? How are they different? They're still going to have a sugar phosphate backbone. So they compare DNA here at the top and then here's RNA. And see, RNA is just one strand. One of the difference is that instead of deoxyribose, it has ribose. Okay, we're going to have uracil instead, and it's usually a single polynucleotide. I say usual because if you study biology further, you're going to see some exceptions when it comes to viruses and things like that. Now, what is the big deal here? Well, this is the central idea in biology. There's some rule. It's viruses actually violate this rule, and but basically the code for a gene is encoded in your DNA, and a gene is information for building a protein, like the protein that makes your hair, or the protein that makes an enzyme to digest starch in our saliva, where we start digesting starch when we eat like a French fry, right? It's found in your DNA, and then that DNA transfers its code to RNA. And then the RNA takes that code to a ribosome to make your protein. Okay? And the ribosomes we're going to study when we cover the cells in a little bit. But you may have heard before, ribosomes are the protein factories, right? So here we go from one nucleic acid in the nucleus. We make our messenger nucleic acid. And that messenger nucleic acid takes the information to the protein, right? So what this is DNA stores the information, and then RNA basically takes that same information to the protein to assemble, to the ribosome to assemble proteins. Okay? So there are some other nucleotides that we see that are important, and they're not, they don't function in themselves individually in the storage or transfer of that information to make proteins. As you can see here, this looks very much like down here in the bottom, this molecule here looks very much like a nucleotide. We have our nitrogenous base here, we have our 5-carbon sugar, and then we have a phosphate, but we have 3 phosphates, so it's a triphosphate, not just one phosphate. And this individual molecule by itself is basically the energy currency of the cell. It's used for energy. You might have heard of it before. It's called ATP, or adenosine triphosphate, and it's adenosine because the base is adenine. Okay, and when we bond adenine with our sugar, which is a ribose, and we know it's a ribose because look at the number 2 spot. It's got the oxygen there. It's not deoxyribose, it's ribose, right? So the adenosine triphosphate. So this part of the molecule is adenosine with the 3 phosphates. And the bonds that are between the phosphates, okay? These bonds right here have a lot of energy. So when we break those bonds, that allows us to do the work in cells that are required to build things, to energize muscles and power them, for us to move, for a little microscopic organism like a paramecium, which has these little hair-like structures. They use this ATP energy to swim around. So it's a universal energy currency. And then there's another case where we have molecules themselves that are going to carry energetic electrons. And there's a mistake here on the slide. It's not FAD plus, it's just FAD. And these are actually nucleotides, and the D stands for dinucleotide. And what these do is, when we're talking about within a cell, when the cell is burning your energy nutrients to generate ATP, these molecules are going to take electrons from that food, energetic electrons, so they're going to be referred to as carriers. So they're going to carry these energetic electrons to your mitochondria, a specific location of the mitochondria, and then use the energy in those electrons to power the production of more ATP. It's really pretty cool the way that happens. But they're kind of storing chemical energy with those electrons, and it's kind of like when you store, where you take that energy and then you're going to use, it's like potential energy in there. And so the way this works here is, this one stands for nicotinamide adenine dinucleotide. It has overall, a molecule has a plus charge to it. And what it's going to do is it's going to pick up 2 electrons, 2 negative charges from the food molecule, and it's going to pick up 1 proton from the food. And so the key here, so remember these are coming from the food, the proton and the 2 electrons. And if we look at, we do the math here, this had a +1 charge on it, the NAD+. The proton has a plus charge on it, and then the 2 negative electrons. So altogether, those items, if we add them up, the charge ends up balancing off. And so the 2 electrons get added, and that 1 proton gets added, and you get NADH. And because the electrons got added onto the NAD now has been reduced. We talked about oxidation and reduction in the prior chapter. So this is the reduced form, and it's carrying those electrons. Any molecule that receives those electrons, we say is reduced. This is the oxidized form of it. Okay. Now, what is that NADH going to do? It's going to take those electrons, the proton goes along with it, to the mitochondria, and then deliver those electrons to help power the production of ATP. There's another one called FAD. It's also a dinucleotide, flavin adenine dinucleotide, and it's FAD only. And so it actually picks up from the food. It's going to, and this has the help of enzymes. It's going to pick up 2 electrons, but it doesn't have a positive charge. It's going to end up picking up 2 protons. Okay. And then that gives us FADH2. And this molecule is the reduced form because it picked up the electrons. And again, where did the electrons come from? From the food. So basically, we oxidize food and reduce these electron carriers. So this is the oxidized form. So what we're going to do is we're going to take electrons from the food, which these come from the food, and then move them onto, or with a plus sign, we're adding them onto the carrier there. And now this carrier is charged up, ready to go. It's reduced, it's ready to go take it. So I found this just to show this. This is a chemical compound found within the cells going through this process of energy metabolism. And don't memorize it, just as an example here. There's your succinate. And then we can see here that there are hydrogens on there. There's 4 of them. Okay, and then here is your oxidized form of FAD, and the FAD is brought in and combined with this molecule here with the help of an enzyme. Okay, and the enzyme is going to transfer 2 hydrogens from there and some electrons, and you end up getting this product here. And then if you look at the result of what this molecule, the succinate, was, and you come over here and you look at it here, and you look and you count the number of hydrogens on there, it's missing 2. This fumarate has 2 less hydrogens. That's because basically we took them off of this organic molecule that had those energy-rich electrons associated with the hydrogen. So, guys, if you're going to reduce one item, you're going to have to oxidize the other. So we're reducing here, this is the reduced form. When we reduce this FAD, we had to have oxidized something else. And what are we oxidizing? You're oxidizing the food. So this is basically coming from your food. That's the way this energy transfer process works. More on that later, but for now, it's just a little bit of explanation of how that works, right, and connecting it to oxidation reduction from our prior chapter. But for now, just be familiar with NAD plus and FAD being energy nucleotides. They're not associated with storing genetic information to build proteins. These carry electrons to the mitochondria for the production of ATP. And when we go back to the molecule, the ultimate goal is to produce this ATP molecule that we see right here. And so it's about transferring these electrons from one to another until we end up building this ATP molecules here.